Okay, I would like to talk a little bit about the issues with synthesis of DNA. Many of you here have heard of sequencing DNA. That's kind of the classic human genome type project. Sequencing is reading DNA molecules. You know, you're given a test tube of stuff, you want to get a string out of it at the end to put in a computer, the sequence of A, G, C, and T that will tell you what it is. Synthesis is kind of the inverse operation. It's about writing molecules to specification. You have in a computer a string on A, C, G, and D, and you'd like to somehow make it molecules that have exactly that pattern. And there is an emerging field called synthetic biology, which is motivated by the fact that technologies for synthesizing large DNA sequences, the cost of that is rapidly dropping. For about 20 cents a base, okay, for virus length sequences, you can synthesize a virus to specification. That means a few $1,000s to make a virus to your specification. And this opens up all kinds of different possibilities. Large scale synthesis, will change the way molecular biology is done because it makes it easy to change things and see what happens. So why me in synthetic biology? Don't be fooled by the man in the coat. I'm a computer scientist. I have never done anything useful in a lab. But that said, designing sequences under constraints is an algorithmic optimization problem. And as an algorithms person, that's the kind of thing that I want to do. So I'm going to tell you a story of a research project that goes through many things here. We're going to told you a little bit about what synthetic biology is. I'm going to review what gene translation is. Tell you a little bit about how vaccines get created. And then to start to discuss an approach we used to design vaccines that works by using evolution against itself. And we'll learn a little bit about evolution and some of the properties of sequences and translation. And then we'll talk a little bit about combinatorial algorithms for optimization. How do you design the best sequence given certain constraints? And we'll review a couple of classic algorithms like dynamic programming and simulated annealing to go through that. And finally, maybe do some tests to see whether this actually works. So the central dogma in molecular biology is that DNA goes to RNA, goes to protein. Biological systems are made out of proteins. To make proteins, you describe that by a DNA sequence, a gene will describe how you go about making that protein. And the way that it goes through and does that description is something called the triplet code. The triplet code, is in some sense a table built into the cellular machinery that maps triples of bases to one of the 20 amino acids that proteins are made up on. And so every amino acid is represented by at least one triple or codon that represents it. Some amino acids are represented by 6 possible amino acids, 6 possible codons. And that these are all, in principle, in some sense, synonymous. You can encode for any protein that you want. A protein is a chain of amino acids. You can code for it by picking any one of the synonymous codons at every position to describe how to make that protein. So that's basically more or less how encoding works on the genetic level. What are vaccines and how are they made, or at least traditionally live vaccines? Live vaccines, or vaccines are something you want to inject someone with a vaccine to stimulate their immune system to respond to a disease-causing agent. You want something that will cause a reaction in your body that will be helpful when you're exposed to the real immune agent. But you also want it to not cause such a response that it'll kill you or hurt you. You would like to have in some sense, if you want to create a live vaccine, you want to create a virus that you can be exposed to, that your body can easily fight off without causing disease. And yet, after having fought it off, your immune system is now trained to recognize the wild type agent, the disease-causing agent, and can fight that back and do it. So traditionally, live vaccines were developed by a process of evolution through animals. You have a disease, human-caused something like polio, that was a disease in humans. You expose monkeys to it. You try to evolve this vaccine, this virus, so it grows well in monkeys. As it grows better and better in monkeys, as you pass it through monkey after monkey, eventually it gets good at growing in monkeys and less good at growing in people. And at the end, what you hope to have is a live virus that will still grow just a tiny bit in humans, but not enough to cause the disease. This does work, but it's very expensive. It uses a lot of money, it uses a lot of monkeys, it uses a lot of time. So we're instead faced with the question of how can we create a vaccine, in some sense, for a new pathogen that emerges. You've probably seen these movies, these science fiction movies, where a virus comes from outer space and starts to wipe out life on the planet. How can we try to build a vaccine rapidly for some new thing? We don't have time to passage it through all these monkeys. We proposed an approach using this fact that we can now synthesize viruses on demand, which we call synthetic attenuated virus engineering, or SAVE. And it's designed for the fact that viral diseases do emerge. You know, Ebola was the rage a few months ago, a few years ago. How might you hope to design a vaccine quickly against such a pathogen? Well, in our method, we are assuming that as input, we are taking the genome sequence of a virus from a database like GenBank. We assume we have the genome sequence. The output of our design process is going to be a design of a virus that is going to have the property that it's going to have exactly the same protein sequence as the original virus, so it's going to work in some sense in the same way. But we're going to engineer the coding sequence in a way that we think it's going to somehow translate worse. Put in a lot of things into the sequence that will maybe cause it to translate worse, translate a little slower, translate somehow less effectively, so that the result is that it will grow less quickly in your body will be able to fight it off, and it will be a safe virus. Okay, so we're looking for something that is going to basically try to hurt viruses by 1,000 cuts, 1,000 little changes, with the hope that it will translate much slower and be something safer. So what is the source of our little cuts? Okay, we are going to use a phenomena that is an interesting phenomena called codon pair bias. Okay, we said that for the leucine amino acid that there are 6 different codons that code for it. For arginine, there are 6 different codons that code for it. That means that there are 36 different possible pairings that could occur in a genome of a way to code for a leucine against a way to code for an arginine. And if you do statistics on the human genome, some of these pairs are represented far less often than you would expect under an independence assumption. There is some reason why the CUU-AGG sequence seems to be dramatically underrepresented in humans. And we're going to go under the assumption that why is it underrepresented? Presumably, evolution is selecting against it. There is some reason why this sequence at the translation level is bad for the way translation works, or something works, okay? And these patterns have been systematically removed by natural selection. Our idea to design a vaccine, we want to design a weak virus. So if we want to design a weak virus to serve as a vaccine, we want to put in a lot of patterns that nature seems to be selecting against. You know, the enemy of our enemy is going to be our friend, okay? And so we want to design a virus that has as many of the underrepresented pairs as possible while coding for the original protein sequence, okay? And our vision would be this should be a weaker virus. Now, to design the optimal sequence under these constraints, we have to realize that there are a lot of different genes that can code for the same protein. In general, if you've got n amino acids, okay, a sequence of length n amino acids, there are about 3 to the n different gene sequences that could code for that protein. And for reasonable sized proteins, this quickly gets to be larger than the number of atoms in the universe. If you really want to find the best possible design, the one that by our criteria of maximizing these bad pairs, okay, you need an efficient algorithm if you're going to design a bug that does that. So how might we go about designing such a thing? Well, there is a classical algorithm that appears in a lot of different places in computational biology called dynamic programming. And the way it sort of appears, again, there's two ways to look at it. The simple, if you know what you're doing, but scary if you don't, way of looking at it is by a recursive formula, okay? And we're going to say that the best possible encoding of the first I bases, amino acid residues of my protein, ending in a particular codon C, well, the best possible way to do it would be, it's going to end in, if it's ending in C, the previous one is going to be some codon D. The cost of that codon, having C and D next to each other, is a function of how much they like each other, okay? And the cost of the whole thing is the last transition plus the cost of encoding, the best possible way to encode the first I-1 residues, or amino acids, ending in D. And if you try this over all possible ending points of the next to last spot, that tells you the optimal way to encode the whole thing. And this is kind of the idea behind dynamic programming, one of these recurrences. Another way to think about it is as a shortest path problem. You could think of a network, not sort of a network or a path, where you have a stack of all possible codons that code for this amino acid, a stack of proteins that code for this amino acid, a stack of codons that code for this amino acid. And you can have a network of edges telling you what the cost is of coding from going from this codon to that codon. And if you see it that way, it's really the best possible design is just a shortest path through this network. And in fact, this dynamic programming thing that looks scary is really just a way of finding a shortest path. And so if you know about dynamic programming, the shortest path thing, sometimes hear it called the Viterbi algorithm. But that's one way that we could design genes. Now, in practice, we don't really use that, at least when we work with our friends, the virologists, okay? In fact, one problem is that when you do a design like that, that will come up with the one that, the sequence that optimizes the codon pair score, but it drastically changes the codon usage. It may decide that it's going to use all of one codon and not of another. And for various reasons we don't want to do, we want to isolate the effect of this. So what we would really like to do is to design a gene that gets a great codon pair score by moving around the codons rather than picking a different one for each spot. The amino acid sequence is going to be the same each time, but we're going to have the freedom to rearrange synonymous codons so that it still codes for the protein, it uses exactly the same set of codons, but it will achieve a worse score for us, okay? And that's what we do. And finding the optimal arrangement of these codons then becomes something that smells a lot like the traveling salesman problem in computer science, where you want to visit all the codons in the cheapest possible way, depending upon the travel costs from one codon to another. And this is a famous and classic hard problem, okay? And so there are heuristics, something like called simulated annealing, that are good at coming up with reasonable solutions to these hard problems. Basically, we randomly swap codons if they're synonymous and see if it does something better. If it does something better, that's great. Ka-chung, we're going to take it and it moves us closer to where we want to be. But occasionally we'll take backwards moves if we swap them and it's not so bad to give us some way of avoiding getting trapped in local optima. So if you do this kind of thing, you can produce designs that are very, very good, close to optimal. How good? Well, if we take a look at this, this is sort of showing an illustration of when we were designing a flu vaccine. Here are three flu genes in codon pair space. Okay, and if you look at that, they are, roughly over here. When we finished rearranging the codons and optimizing them, they are way out here. They look like genes that don't look like things that tend to get existing naturally. So we make our flu vaccine, and what happens? Again, to know what really happens, you got to shoot it in an animal. And, to summarize the data quickly, because I can see I'm running a little low on time, if you take our vaccine, it's weak enough that you can inject it into the mice and they basically stay as healthy as if you had just injected them with water. So it's not really a virulent thing. But when you actually now take those mice and expose it to what should be a lethal dose of the flu, all of our mice survive, including to a pretty big safety range before you can do harm to our mice. And working on this, other groups have taken on this idea. So you can, in principle, this is an idea that seems like it could be applied to lots and lots of different kinds of viruses. It's attacking something very fundamental, okay? And we see people with other viruses doing this kind of thing. And so this is a banner moment for us at Stony Brook. So to conclude, I'd just like to say my take-home message is that DNA synthesis costs are declining, and this opens up all kinds of exciting things. And taking advantage of these things involves really some fundamental computer science and algorithms to design the optimal thing. And these are my, on various projects, these are my collaborators and students, and that's pretty much what I want to say. Yes?
Do you have some homework assignments in mind for us to do?
Well, if you have a homework assignment, so if you want to do something, what would seem like a fun thing to do would be, given such a cost matrix, have people design their own genes and see what actually happens. To what extent could you, does this kind of dynamic programming algorithm actually produce an interesting gene? And when you compare that to what you would get by swapping the codons around, how well does a heuristic work? So these seem to be on a computer sciencey level, interesting things to play around with. And then you can start to say, well, gee, there's this interesting scoring system. How is it that real genes score? Do real genes care about these codon pair biases or not? What's causing them? That seems like a perfectly good programming exercise for an elementary biology class. Yeah.
So I was wondering why you abandoned the first path of finding the shortest path and sort of the optimal, seemingly naive problem, and then said you want to keep the same set of codons but rearrange their order. So I guess it's in two parts. Why did you do this in the first place? And second, in what sense is this new idea optimal, or is the real answer to do someplace in between?
Okay, so the real reason we want to do it is partially aesthetic, okay, and partially scientific. So first of all, it should seem weird to certain biologists, I don't know how weird it will seem to you, that I can take the same codons and shuffle them and order them in a way that I can almost certainly knock the gene down, okay? This shouldn't, by your zeroth order model of how translation works, this shouldn't happen. So this phenomena of codon pair bias is a new and interesting thing. And if you want to study this, which our group is interested in, is studying what is the effect of codon pair bias on translation, you need to have controls. And controls would be, nature's sequence, our sequence. We haven't changed codons. You think changing codons is important, okay? We've shown that there are other things that are important. Earlier, we had a talk by Carl Kingsford sort of showing you, changing the codons does change the translation speed, and tried to quantify how much that is. We've done similar work trying to figure this stuff out, but you find that there's relatively small effects of translation speed. The difference between a high speed and a low speed thing is fairly slow, fairly small. And so to shed light on this whole phenomena, okay, this is why we sort of chose to do this. Okay, fair enough. Oh, yes, wait, so not so fast, yes.
Do you have any insight then as to what might be the causative agent behind this slowdown? Or is this still an open question? Are you working on it or others working on it?
This isn't the question is, again, why does this work? So the cool thing about this is on one level, we don't need to know how it works. We're just putting in things. You tell me, I look at this, I see what's bad. We put it in, it works, okay? But as scientists, we want to know why does this work? And our first assumption was that it had something to do with the speed of translation with codons and stuff like that. And, again, we did this kind of analysis, somewhat similar to what Carl did. And it doesn't seem to be as big a difference as you would think. A bigger thing that seems to be happening is the amount of RNA is being rapidly degraded. Okay, so there seems to be an effect that this is due to degradation of RNA. That it's not like the messenger RNAs are not, it's not that they're just being translated slower. somehow they're getting eaten up for various reasons. And we have some reasons to believe why we think that that is. Okay, but this is something I have collaborators in microbiology, and we talk yeast every week, and this is one of the things we're working on.